Hello everyone, in the last video we discussed about membrane potential. We discussed in detail how action potential and resting potential is induced in nerve cells or neurons. Today we are going to discuss how the information is transferred from one neuron to the other neuron. Neurons or nerve cells. These are specialized cells that respond to stimulus and convert it into action potential. Neurons communicate at the meeting point and this is known as synapses, and the information or the signal flows through these synapses in only one direction. There are two types of synapses: chemical synapses second, electrical synapses. Electrical synapses is also known as gap junction. The flow of information through this electrical synapses is rapid compared to chemical synapses. In this video, we will focus on chemical synapses. Space between the presynaptic and postsynaptic neuron is known as synaptic cleft. In this presynaptic terminal, membrane-bound vesicles are present, that is known as the synaptic vesicles, and these are filled with the neurotransmitters which acts as the chemical messengers. When action potential reaches the presynaptic terminal, then it causes the voltage-gated calcium channel to open. This results in the influx of the calcium ion, as a result of which the concentration of calcium ion increases in the cytoplasm of presynaptic terminal. When these synaptic vesicles fuses with the plasma membrane, neurotransmitters are released in the synaptic cleft by the process of exocytosis. Then these neurotransmitters diffuses across the synaptic cleft and binds with the receptors that are present in the postsynaptic terminal. When these neurotransmitters or chemical messengers bind with the receptors present on the postsynaptic terminal, then this results in the opening of the ion channels, sometimes closure of the channels, as a result of which ion flow either inside or outside. This results in the change in the membrane potential of the postsynaptic terminal. Action potential is induced. In this way, the information flows from one neuron to the other neuron. Let us understand the transfer of information from one neuron to another neuron once again. This is presynaptic neuron, this is postsynaptic neuron. These communicate at the meeting point known as synapses. The space between the presynaptic and postsynaptic neuron is known as synaptic cleft. When the action potential reaches to the terminal of this presynaptic neuron, then there is the change in the membrane potential, as a result of which voltage-gated calcium channel opens and there is the influx of the calcium ion, as a result of which the concentration of the calcium ion in the cytoplasm increases. This results in the fusion of synaptic vesicles to the plasma membrane of the presynaptic terminal. And by the process of exocytosis, neurotransmitters present in these synaptic vesicles are released in the synaptic cleft. Then these neurotransmitters diffuses across the synaptic cleft and binds to the receptors of the postsynaptic neuron present at the terminal. Binding of this receptor causes the opening of the ion channel present in the plasma membrane of postsynaptic terminal. Sometimes it closes, as a result of which ion flow inside or outside this channel. Hence, the membrane potential of this postsynaptic terminal is altered and action potential is induced. In this way, information flows from one neuron to the other neuron. Types of chemical synapses: excitatory or inhibitory. Binding of neurotransmitters to the receptors either promotes or inhibits the generation of action potential in the postsynaptic neuron. When the neurotransmitters bind to the excitatory receptor present on the postsynaptic neuron, then it leads to the opening of ion channels that causes the depolarization of the plasma membrane of postsynaptic neuron, and this promotes the generation of action potential in the postsynaptic neuron. And when the neurotransmitters bind to the inhibitory receptor present on the postsynaptic neuron, then it causes the opening of the ion channels, which causes the hyperpolarization of the plasma membrane of postsynaptic neuron. And this inhibits the generation of action potential in the postsynaptic neuron. Neurotransmitters binds to its specific receptor by lock and key mechanism. That means these neurotransmitters bind to its specific receptor, like lock has its specific key. Acetylcholine and acetylcholine receptor. Acetylcholine is a neurotransmitter that produces both excitatory and inhibitory response. This is because of this acetylcholine receptor. There are two types of acetylcholine receptor: nicotinic acetylcholine receptor, which is excitatory receptor, and muscarinic acetylcholine receptor, which is inhibitory receptor. Let us see this nicotinic acetylcholine receptor. This is presynaptic neuron. Acetylcholine is released in the synaptic cleft. This then diffuses and binds with the nicotinic acetylcholine receptor present in the membrane of the muscle cell. This is ligand-gated ion channel. Binding of this acetylcholine results in the opening of the ion channel. This allows the movement of both sodium and potassium ion. Sodium moves inside and potassium ion moves outside. Because the gradient of sodium ion across the membrane is steeper compared to the potassium ion, so the inward movement of sodium ion exceeds the potassium ion. This results in the localized depolarization of the membrane of muscle cell, and this triggers voltage-gated sodium channel to open, as a result of which more sodium ion moves inside, and this leads to the generation of action potential. Snake neurotoxin, example alpha bungarotoxin and cobra toxin, binds with the nicotinic acetylcholine receptor and thus blocks the transmission of impulses between neurons and muscle cells. Now the muscarinic receptor, that is inhibitory receptor, it is transmembrane protein. When acetylcholine, that is the neurotransmitter, binds with this muscarinic receptor, it leads to the opening of voltage-gated potassium channel, as a result of which efflux of potassium ion takes place. This results in the hyperpolarization of plasma membrane, so no action potential is generated. Atropine, that is obtained from Atropa belladonna, when it binds with muscarinic receptor, it inhibits its function and it is used for pupil dilation. So this is all for chemical synapses. In the next video, we will start with new topic, cell cycle. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.